Hello everyone, welcome to my channel Bunny's Biology. Today we will learn about one of the most powerful defense system our cell used to protect the genome, that is nucleotide excision repair, also called NER. So DNA is constantly attacked by UV ray, environmental mutagen, certain chemotherapeutic agent and free radicals, all of which causes lesion in DNA. And this distortion bend the DNA helix and block the vital process like replication and transcription. Nucleotide excision repair pathway targets and remove bulky lesion from the DNA helix. Without this repair pathway, cell would accumulate mutation and leading to disease like cancer. Here the UVA, UVB radiation that causes pyrimidine dimer and oxidatively generated damage. So in this lecture, we will explore NER step by step. NER in E. coli is largely accomplished by 4 proteins, that is UVR A, B, C and D protein, and that is encoded by the gene UVR A, B, C and D gene. So in bacteria, the complex of 2 UVR A and 2 UVR B molecule first scan the DNA where the distortion or lesion present, with the UVR A being responsible for the detecting the distortion in the helix. And this ATP hydrolysis promote dimer formation between UVR A and UVR B. Upon encountering a distortion, UVR A first exit and then remaining UVRB hydrolyzes the hydrogen bond present in two DNA strand and create a single stranded bubble around them. This is the bubble formation. Then UVRB recruit UVRC and UVRC create two cart. One located 4 to 5. This is the 1, 2, 3, 4. 4 to 5 nucleotide 3' to the lesion and another 8 nucleotide 5' to the lesion. The lesions containing strand is removed from the left of the DNA. This is the removed DNA with lesion. This is the lesion by the DNA helicase UVRD. So resulting 12 to 13 nucleotide long gap. Finally DNA polymerase and DNA ligase fill the gap. This is the mechanism of E. coli. But in higher cell, the NER repair much the same as that of E. coli. In higher cell, XPG, which is responsible for the detecting the distortion to the helix as like UVRA. So here E. coli UVRA first detecting the lesion that present in DNA, and that is equivalent to XPC in higher cell. That first detect the lesion and then the DNA is open to create a bubble around the lesion. The formation of bubble involves the helicase activity of the protein like XPA and XPD, that is equivalent to UVRB in E. coli. And XP and XPD in higher cell. So first XPC detect the lesion and then XPA and XPD form the bubble. Then this bubble formation create cleavage site. One is 5' to lesion, that is cut by the nuclease ERCC1 XPF, that is 5' to lesion, and another is 3' to lesion, that is cut by nuclease XPG. So here ERCC1, XPF and XPG that cut the DNA, and that is equivalent to UVRC, that is equivalent to. So in higher cell, the resulting DNA strand 24 to 32 nucleotide long is removed and create a long gap, and this is filled by the DNA pol 1, DNA polymerase 1 and DNA ligase. So thus NER serves as a critical safeguard against mutagenesis and ensuring that damaged DNA is replaced with a clean copy to keep the genome stable. So if any defect in nucleotide excision repair mechanism that causes the disease like Xeroderma Pigmentosum, where patients are extremely sensitive to sunlight and prone to skin cancer.